Hi everyone, good evening. This is Neeraja. I'm a senior genetic counselor and I'm here to introduce you to a new topic of our ORDI webinar, which is human-based complex in vitro models. It's very exciting to see the combination of both rare diseases and therapeutics and discuss this today as one of our potential promises and potential therapies in rare diseases. I have with me Dr. Surat, who is an expert in this field. She has done her PhD from Tata Institute of Fundamental Research, TIFR, and she has a lot of experience in this area. Currently, she works as a senior program director, research and toxicology at the Humane World. And across various sectors, she works with both biomedical research, chemical, agrochemical safety testing, vaccine, batch safety testing, and drug development, and works with multiple stakeholders such as regulators, policymakers, industry, and academia. Very exciting to have you here, Dr. Surat, and we would love to hear your insights on where we are going forward in the field of rare diseases and therapeutics today. Welcome.
Thanks, Neeraja, for the kind introduction. And I'll just share my screen. And thank you for the opportunity. 1 second. Just let me know if you're able to see the screen.
Yes, and if you can just put that in full screen.
Yeah. Good evening, everyone. And first of all, thank you, ORDI, for giving me the opportunity to speak on this topic. And today I'll be talking about what is the potential of complex in vitro models for rare disease therapeutics. And like it was mentioned, I'm currently working at Humane World for Animals India, and we work across different sectors of biomedical research, chemicals, vaccines, to see how we can enable the use of emerging human-based technologies in contrast to animal models. So before I get into the actual content of the presentation, I just want to briefly talk about our organization. So Humane World has a research and toxicology program which is spread across more than 50 countries, and we work with different stakeholders such as industry, regulators, lawmakers, researchers, and our team includes experts in different areas such as regulatory science, biomedical research, science communication, etc. And we work towards enabling the use of human-based technologies and replacing the use of animal testing in both research and testing space. And our approach has been a multi-pronged method where we work towards how can we increase funding to develop these newer technologies, because these newer methodologies are still emerging. So it requires strategic and dedicated funding to develop these systems. At the same time, we also need to work towards increasing the capacity and confidence in these newer technologies. Then, apart from development, unless these technologies have regulatory approval and acceptance, they cannot be adopted widely. So how can we work towards the regulatory acceptance of these newer methodologies and modernize laws and regulations to see they are globally harmonized across the globe? So these are some of the methodologies by which we work. And specifically with respect to Humane World for Animals India, we are a non-profit company. And apart from research and toxicology, so that is one department, we also work across other spaces such as farm animal, wildlife, street dogs, disaster response, and cruelty response situations. So that was about the organization. And then broadly coming to the topic of our discussion today. So my talk will be broadly divided into these 4 areas. So first is I would discuss why do we even need human relevant models for rare disease therapeutics? Why are we talking about this? Second, I would just discuss some case studies of how these models are being used during early drug development and discovery stages. Then, apart from the research space, I will discuss if there are any regulatory submissions that have been made using these models for efficacy and safety. And then I will end with what kind of recommendations that we can work towards to establish these pathways and accelerate this field in India. So coming to the first point, like why do we even need human relevant models? So this kind of boils down to what is the current pipeline of drug development. So I'm not sure how many of you are aware, like for example, the current drug development pipeline, for every 10 candidates that enter the clinical trial stage from the preclinical testing phase, almost 9 of them don't make it through, and only like one finally receives regulatory approval. So there is almost 85 to 90% attrition rate of the targets that enter the pipeline. So, and this is this is an extremely leaky pipeline, which as you can see, and there have been several studies which have documented that, and this is kind of a known fact now of how leaky this entire drug development pipeline is. And in terms of how inefficient this is, and a recent study has also looked at more recent data also, where they looked at 18 leading pharmaceutical companies, and from 2006 to 2022, to see again how many candidates actually receive approval, in terms of like how many candidates enter the drug development, like the clinical trial phases, how many of them actually receive approval. Again, the percentage was very low. It's only around like 14% of candidates that finally receive the approval, which is basically the regulatory approval, and enter the market. So, and this is one of the reasons why this one of the reasons for this inefficiency is also because of differences in translation, because the preclinical stages mainly rely, conventionally they have relied on animal models, and like we do know that there are vast differences in animal biology and human biology, and how animals versus humans respond to diseases and drugs. So there are significant biological differences. I'm just giving like some examples in the context of rare diseases, but this is not specific to rare disease. I mean, this is a general drug development problem, you could say, but specifically in the context of cystic fibrosis, for example, which is a rare disease. So in humans, cystic fibrosis is associated with chronic infection, inflammation, airway remodeling, and mucus obstruction, whereas the mice models of cystic fibrosis have normal lung function. And this is true for many rare diseases. Like the animal models often fail to replicate what is observed in humans. Another a very popular example of this lost in translation path is like a monoclonal antibody trial which happened in 2006 in London, and this trial and this antibody was found to be completely safe in non-human primates, but then it triggered when it was ultimately tested in humans at doses which were almost 500 times lower than what was tested on humans. What was tested on animals, it led to like massive life-threatening reactions and multi-organ failure, and ultimately the trial was stopped because of obvious reasons. But then all of these are examples of how in many cases, like what is observed in terms of safety and efficacy in animals doesn't translate to animal model, doesn't translate to humans. And this is one of the reasons why there is a high attrition rate, because like the targets enter the clinical trials based on the preclinical data in animals. So this kind of indicates that why we need newer systems which are more relevant to humans, which are based on human biology. So these models were developed when we didn't have a lot of the technological advances that have happened in the last few decades. So we also need to kind of advance with the newer technologies that have come up, which are based on human biology, to have like more efficient drug development pipelines. So what exactly are human relevant systems? So another question which many people are often unclear about, like if we don't use animals, what exactly do we use? So there are currently now many newer systems which have been developed. So there are some examples of these I have are shown here. So these are in vitro technologies which are based on human stem cells or human primary cells. So these are called complex in vitro models. So the conventionally used in vitro model was a 2D cell culture model where the cells are grown flat on a two-dimensional Petri plate. But that 2D cell model is also not very replicative of human biology, because inside our body, cells are not flat cells. They are three-dimensional structures which are exposed to physical cues, mechanical cues, biochemical cues. So the conventional 2D in vitro model is also not very replicative of human biology. So these newer systems which are shown here, they are called complex in vitro models, which are three-dimensional, and are they replicate like certain aspects of human biology and human organs in a much better manner than the conventional 2D system or animal model. So some examples of these are spheroids. Then there are organoids, which are um, miniature versions of organs. So for example, there are brain organoids, there are liver organoids. So they are not exactly similar to a brain, like a complete brain, but they mimic certain elements of brain, functional elements and structural elements of brain. Similarly, there are liver organoids, kidney organoids, and so on and so forth. Then another such model is organ on chip. So these are AA battery sized microfluidic systems which are aligned with human cells. And again, they mimic certain elements of human physiology uh, um, in a much better manner compared to like the two-dimensional cultures. And then we also have now 3D printed tissues. And similarly, there are also several advancements in silico tools now, but today I will not be going into the in silico tools. But then there have been like several advances also in this space. So these are some of the systems which have been developed in the past decade, and which can mimic the human biology to a great extent, or certain aspects of human biology to a great extent. So, and there has been a global shift towards how can we increase the use, adoption, and development of these models. So globally, countries are recognizing that we need to have model systems that are more predictive of human biology. And towards this, these are again just some examples of all the global work that has been happening in this space. Like US FDA had a roadmap to reduce animal testing and move towards these new methodologies in preclinical safety studies. Recently, just 2 months back, US FDA also had a guidance document on how to use these new approach methodologies in drug development. So that's what these methods are called. They're called like new approach methodologies, which can be like either uh, a single complex in vitro model or a combination of uh, different complex in vitro models, or a combination of complex in vitro plus in silico models. So yeah, so these are called new approach methodologies. And CEDAR, which is the Center for Drug Evaluation and Research, so that also came up with a documented list of what are the acceptable new approach methodologies that can be used for regulatory submissions. And National Institute of Health at US, NIH, also recently gave out a statement that they are going to prioritize human-based technologies. And in fact, even in terms of funding, they said that all the funds that would be released from NIH, they would be for projects which have a human biology based model. And if there are any projects that are based only on animal biology, so those kind of projects would not be funded anymore. So that's a very strong statement from a public funding body to create an incentive for developing these systems at an accelerated pace. Similarly, there have been recently UK government has also come up with a roadmap as to how to develop a strategy to support the development, validation, and uptake of these alternative methods. Similarly, EU is also in the process of developing a roadmap for chemical safety testing. And in the European research area policy agenda for 2025-27, so they have clearly marked as accelerating the new approach methodologies as one of the priority areas to advance the biomedical research and testing of medical products and devices. So you can see all the global shift which is happening to accelerate the advancement and development of these methods. Even in India, a lot of changes have been happening, although it is at relatively nascent phase compared to the global scenario, but still it's not that India is not immune to all these global shifts which is happening. So again, these are just some examples. In 2018 itself, ICMR had come up with a white paper to how we can develop these methodologies in India, under the leadership of Dr. Balram Bhargav. And then under Dr. Soumya Swaminathan, also ICMR developed a perspectives paper as to how we can develop this space in India. Recently, BIRAC, DBT BIRAC has also been very active. Like last year, they conducted a stakeholder consultation as to how we can have regulatory pathways to develop these complex in vitro models. And there was also a workshop to develop funding mechanisms to develop these methods in India in 2024 in collaboration between Humane World, BIRAC, and Office of PSA. CDSA has also come up with several regulatory initiatives. Like there was an amendment to the NDCT rules in 2023, which allowed the use of these methods, such as cell-based methods, organ-on-chip, and other human biology-based methods to test for safety and efficacy of drugs. And recently, the Bureau of Indian Standards, the BIS, has also formed a working group, an organoids and microphysiological systems working group to develop national standards for organoids and organ-on-chip methods. So a lot of efforts are also happening in India, but we are still at a relatively nascent stage compared to the global scenario. So coming to now, I also want to give some real life examples of how these systems are being used for drug development. So I will talk about both early drug development stages, like in the biomedical research space and also in the regulatory submission space. So coming to case studies where these systems are being used to understand disease mechanisms or in the drug develop initial stages of drug development stages. So for example, one example has been in the case of DMD, which is associated with progressive skeletal muscle and degeneration. And what this study did was they took patient derived, like DMD patient derived induced pluripotent stem cells. And subsequently, they created skeletal muscle organoids from the stem cells that were taken from the DMD patients. And they were able to mimic some of the phenotypes that are observed in the DMD patients. Like, for example, there was developmental delay in the skeletal muscle organoids, which were grown in the lab. And similarly, if we take another case, like this another rare disease, which is the spinal muscular atrophy, SMA. And again, in this case, what the researchers did, where they took patient derived organoids. So by patient derived organoids, I mean they basically take stem cells or stem cells from the patients, and then they differentiate it to form specific, like for example, they were patient derived human induced pluripotent lines, and then subsequently they formed neuromuscular organoids from those patient derived lines. And then and they found that the organoids that were created from the patient derived lines, they exhibited neurodevelopmental defects compared to the controls. The controls would be, like for example, a normal patient, a normal human. So there were specific phenotypes that were observed in these organoids, which were derived from patients compared to the normal phenotype. So these organoids were able to mimic some of the features or phenotypes that were observed in the disease stages in the organoid. Which shows that this system can be used to identify new drug targets and understand how exactly this what exactly is the mechanism behind these diseases. Similarly, another disease where a lot of work is happening is cystic fibrosis. And again, in cystic fibrosis, there are several labs that have also created cystic fibrosis on a chip, which is basically, it's again a microfluidic chip, which is basically has like microfluidic channels, which are lined by the bronchial cells from, for example, it could be patient derived. And and it was found that this cystic fibrosis on a chip was able to mimic many features of human cystic fibrosis airways, like which are usually seen. For example, there is enhanced mucus accumulation, there is increased cilia density, and so on and so forth. And there is enhanced inflammation or pro-inflammatory cytokines. So all of these phenotypes were mimicked in this cystic fibrosis airway on a chip. So showing that these systems can mimic the human phenotype to certain aspects of the human phenotype, which is observed in those specific rare diseases. So this was in the like drug development stages. But then apart from this, there are also the regulatory submissions which have been made using complex in vitro model data. So by regulatory submissions, I mean when the data is submitted to the regulatory authorities to get approval to conduct clinical trials. So basically the preclinical data is submitted or the non-clinical data is submitted to the regulators to get approval to conduct or start conducting the human clinical trials. So that is where so those are like the critical stages where the data needs to be um, there it needs to be a very strong data set in order to convince the regulators to start conducting the human clinical trials. So even in those cases, there are instances where the pharma companies are beginning to make use these systems to make the regulatory submissions. One such example has been for a rare disease called CIDP, which is chronic inflammatory demyelinating polyneuropathy. And for this rare disease in 2022, Sanofi filed an IND for this disease for a small molecule based on the efficacy data that was generated purely from a human on chip system. So I just want to kind of point out that for this particular case, the safety data was already there. So it was a repurposed drug. So by repurposed, I mean, so it was, it was a drug which was already being used for another indication. So the safety data was already existing, and the efficacy data was generated using using the complex in vitro model, and using only the complex in vitro model data, the efficacy data was generated, and they could get an approval to conduct the clinical trials based on this data. And right now the clinical trials are ongoing for this, so that is a very promising example. Similarly, recently, like there have been publications which are also trying to understand how many like pharma companies are beginning to use these systems for their regulatory submissions. So again, this was a publication last year which looked at almost 27 to 28 pharma companies and then tried to see like like what kind of model systems they are using, and for what kind of diseases they are basically using these systems. And they found that almost 50% of the pharma companies are beginning to use these models. So they are not relying purely on animal data anymore. They are using both in silico, which is basically the computational models, and human in vitro models, complex in vitro models, or ex vivo models. So they are using these systems to in their regulatory submissions. And the publication also looked at what kind of diseases were they are using these systems for. And the top most disease category was cancer. And apart from cancer, the second category was serious and life threatening diseases. And in that category, rare diseases also featured. So rare disease is also a significant category where the pharma companies are using these systems to make a regulatory decision for taking a target to the clinical trial stage or not. So there is definitely significant uptake of these methods for rare disease drug development globally. And apart from so this was in terms of efficacy. So apart from efficacy, there are also safety studies, which so efficacy is when you're trying to say whether the particular molecule is effective on the in kind of elevating the disease symptoms. But at the same time, you also need to ensure that the target you're developing is safe and does not have any unintended toxic effects on humans. And there is a specific program which has been started by US FDA, which is called the ISTAND program, or the Innovative Science and Technology Approaches for New Drugs, where they are trying to qualify new drug development tools, including these complex in vitro models and in silico models, and how they can be used for assessing the safety of new drug targets. So this has a multi-step pathway. Like for example, initially you have to submit a letter of intent, and then once the letter of intent is approved, then you submit a qualification plan as to exactly how your system is qualified to understand the problem you're trying to investigate. For example, if you're trying to understand whether your molecule has liver toxicity or not, so you're basically trying to understand how well your system is able to assess that liver toxicity, and then ultimately, if it passes the ISTAN program, it is a qualified tool and it can be used for regulatory submissions. So right now there are 13 letter of intents which have been accepted for further processing in this program, and 3 qualification plans have been accepted. So I'm just giving like some examples of what kind of context of use um, these tools are being developed. So the topmost is like predicting liver on chip for predicting drug induced liver injury. So now there are like a few companies which are developing liver on chip, and a company has also shown that liver on chip can predict liver toxicity better than animal studies. So it is basically drug induced liver injury, and drug induced liver injury is one of the main one of the major reasons why a drug is discontinued, like post, sometimes even post approval, because it's often difficult to predict this part in animals. So there are several. So these are some examples of in which spaces these technologies are being developed. So liver toxicity is a major space where a lot of these systems are being developed. But then there are also like kidney on a chip to assess whether your molecule has drug-induced kidney injury. Then there are also organ on chip to understand developmental and reproductive toxicity for new drug candidates. There is also cases for to understand cardiotoxicity, which is also, if your drug molecule can have a cardiotoxicity risk. So these are some of the examples of where these new systems, these new technologies are being developed. So what kind of spaces they are being developed, and they are already in process for in this program, the STAND program. And if they are accepted, so they can be used as official tool for regulatory submissions. So this just to show that what is the current status of the development of these systems. um, and then I will just come to what can we do in India. So a lot of these changes, what I've shown, they are happening globally, and in the end, that's where the world is moving towards. So it is moving towards more human biology based predictive tools, more human cell based methodologies to predict new drugs and new drug modalities. And these emerging technologies are, they are being accelerated, the development and their regulatory adoption is being accelerated globally. So that's where the field is going to be in few years or a decade or so down the line. And in India, if you also want to be a key player in the development of these technologies and in this space, rather than being a passive adopter of these technologies once they are developed, we also need to start developing these systems in India also, and understanding how we can enable the development and use and regulatory approval of these technologies, so that we can also be a significant global player in this space rather than a passive adopter. So so some of the ways that we can do this is have like establish policy incentives and also have like funding pathways, because right now these technologies are not very much developed in India, and we need a lot of dedicated funding to develop these systems in India. So one of the ways could be like identifying what are the priority areas where we need to develop these tools. So, for example, there could be some ways via which we could do the prioritization. For example, in the national policy for rare disease India, they have identified which are the diseases where, for example, the cost of treatment is very high, and um, there are lesser treatment modalities available. So these are areas of like unmet need and urgent need to kind of have treatment strategies to be available in India. So that could be one area how we could like prioritize which diseases to kind of focus on. There are also other prioritization matrix. So one is diseases which are associated with high cost and do not have any treatment strategy right now. And then there are also diseases which are highly prevalent in India. So based on rare disease registry, we do know that the neuromuscular or neurodegenerative disorders, blood related disorders, and inborn errors of metabolism. So these are top three types of rare diseases which are highly prevalent in India. And again, kind of talk about what is the unmet need in India, what is the urgent need in India. Then there are also diseases where certain technological advancements in terms of developing human relevant model systems has is already happening to some extent. For example, cystic fibrosis, Gaucher disease, or Fabry disease, where for some of these diseases, a lot of these human relevant models are already being developed and have a relatively high technology readiness level. So that is another space to, and that's another parameter to consider. And then there are also like what are the diseases which are being extensively investigated in India. So we looked at like in terms of publications, what are the kind of rare diseases that are being intensively researched upon in India. And then so these are some of the diseases in terms of the number of publications. So that include cystic fibrosis being the topmost, and then we have DMD, Gaucher disease, SMA, Pompe, and Fabry disease. So these are purely in terms of the number of publications and the quant of research that is happening in this country. So these are some of the parameters by which we can maybe prioritize what is the urgent, where the urgent need is, where is the unmet need in India, where do we urgently need a therapeutic intervention where which is not being able to achieve via the conventional paradigm. So we can identify some of these priority areas based on all of these parameters, and then work towards work with different stakeholders to have like dedicated funding to develop more human relevant models in this space and have accelerated development of drug development using these human-based model systems. So on a note, I just want to kind of highlight that currently Humane World is developing a roadmap for funding for NAMs. I mean, not just for rare diseases, but in general for new approach methodologies in India across different sectors. When rare diseases definitely a promising and an unmet need in India. So we are definitely focusing on that area. And apart from rare diseases, we are also focusing on other areas such as like, for example, cell and gene therapy, and then biosimilars. So there are other spaces also which have a lot of unmet need and urgent need to develop therapeutics which are specific to India. And another point is that a lot of these systems, they're also based on patient derived stem cells. So that is another point of consideration, because a lot of rare diseases are often like genetic in nature, and they are specific, and Indian population has I mean, there are genetic differences between Indian population and populations globally. So developing these systems which are based on the patient populations in India can give a much precise way of trying to understand what kind of therapy would work, what kind of targets would work for the genetic profile of an patient, of an Indian patient with a specific variant of a rare disease. So we are currently working on a funding roadmap, which with both public, private, and philanthropic stakeholders to see how we can develop a roadmap to develop these technologies in the next 5 years or 3 years and have like a roadmap laid out. So we need to work in a multi-pronged way where we need to develop these technologies, have them accepted regulation wise, also build the capacity and infrastructure to support the development. Because if we need to have like the entire supply chain established in India, because the cost also has to be kept down if we want to make these technologies more widely accessible. So it is going to be an intensive roadmap that we are currently working on, and rare disease definitely features prominently as one of the urgent areas in that roadmap. Apart from that, there are also several policy pathways that we can adopt to accelerate the adoption of these systems. Like, for example, these are just some examples of different policy pathways that have been adopted by countries such as US, Europe, UK, Japan, and China. So, for example, this the first one is the START pilot program, where again, which has been initiated to accelerate the development of novel drugs and helps to kind of handhold the technology developers from the initial stages till the final regulatory submission, because it's a new technology. So there needs to be a constant crosstalk between the regulators and the technology developers. So these are, for example, some of the programs which can enable that early scientific engagement and co-development of these systems with regulators and the technology developers. Similarly, EU also has a task force which they have set up to provide scientific advice on how these tools can be validated and qualified. And they have a separate pipeline for qualification of new methodologies. Similarly, there are some of these other examples. I mean, I'll not get into the detail of each of these programs, but these are all initiatives which can help to work on the path towards the regulatory acceptance of a new technology, because they are not like we have always relied on animal model, which has a certain kind of a paradigm. Because these are new technologies, we are undergoing a paradigm change as to how these technologies will be viewed, how these technologies will be validated. So we need to work very closely with the technology developers, regulators, and industry together, and cross talk intensively to understand how we are going to develop these technologies. And there needs to be policy mechanisms to enable that kind of crosstalk. So these are just some examples. And then finally, I mean, just in terms of what could work in India is in terms of policy pathway, if we can have some kind of policy pathway like START or a rare disease endpoint advancement track, which basically enables constant and early stage interaction between regulators and technology developers and industry to ensure that that final tool which emerges can receive regulatory acceptance and aligns with what the regulators see as a tool that could ultimately help to understand the safety and efficacy. And a lot of times also in the regulatory committees also right now, because these are new technologies, there might not be experts who are well versed to understand how to understand the data that emerges from these newer technologies. So we need to have a scientific advisory committee within the regulatory framework, who can assess and understand and evaluate these newer technologies, and have that kind of capacity and insight to kind of evaluate these new technologies. Similarly, in terms of funding, we need to have a dedicated funding program to develop these methods, and we are definitely working on that front, and hopefully we will work towards that in the coming year. And then we also need to have again funding incentives, where funding can be linked with regulatory milestones, so that finally funding actually leads to some kind of technology which can be used for regulatory submissions. And also other financial incentives can be introduced to incentivize more academics and industry stakeholders to start using and developing these tools during the drug development process, and also specifically for rare disease scenario. So with this, I'll just stop, and I'm happy to take any questions.
Thank you, Dr. Surat, for that wonderful discussion and really insightful discussion on how these crossroads can work together and what are the important priorities right now, especially for the Indian cohort. Gives us a lot of direction. I will open the forum for any questions. You and participants can type in their questions in the chat box and we can take those. Thank you. Another questions I had was from the perspective of the clinicians as well as the patient advocacy and the the families itself, how do you see their roles in this kind of model where we are trying to bring in the development of new therapeutics?
Sorry, your voice was not clear. So can you just repeat that?
What was the roles of families and clinicians in this kind of role where we are trying to bring in people to look more at policy making for therapeutics in rare diseases?
Yeah, I mean, definitely because a lot of these systems are based on patient-derived, stem cells and patient-derived lines. So definitely, I mean, the patient groups have significant role to play, and even in terms of raising, because I think a lot of patient populations and patient groups may not be aware that such options exist. So and a lot of, like, for example, even globally, a lot of development of these systems is also driven via patient groups, or because they also see the value in what this could ultimately, I mean, ultimately they would be the end users of these technologies. So I mean, in many cases, I wouldn't say right now we are at the point where, for example, tomorrow we could use some of these technologies. But if we start, I mean, investing and driving awareness and creating, funding mechanisms to develop these systems, definitely the end users and the beneficiaries of these technologies would be the families and the patient groups. So definitely, I mean, creating that awareness that such technologies exist and how can we work together to push the development of these methods so that ultimately they can be used for regulatory decision making.
Thank you. So one question on the chat box is it going to replace AI models in the future?
So AI models are already being used, but then AI is also as good as the data which feeds it, right? So we need to generate data, and right now, like, for example, the data which is there is from animal models. So you need human based data to drive your AI model also. So unless you have a strong human based data to drive your AI model, so that's how predictive it would be. So I definitely see value in AI. Ultimately, like, if you would create like a predictive framework where you have a molecule, you could probably have like an AI model which could predict whether it's how effective it's going, but it needs to be driven via human based data. It could be like real world data or patient derived data such as these models. So yeah, I see value in both.
Absolutely. Great. All right, so I don't see any there's one more question. What parent support groups can do for rare disease awareness and treatment to
Yeah,
forward rare
so
disease awareness?
right, right. I mean, right now we are also working with, I mean, the plan is to also work with different philanthropic organizations to see how we can accelerate the development of these models, because right now this space is still very nascent in India. So we need to, I mean, accelerate the development of these systems, and how can we, fast track the development of these models. So definitely, like, we can work with the patient parent support group or the patient support groups to work with philanthropists to see, because it's also, I mean, we would have to kind of drive and the development of these systems, and funding is going to be a core element of that. So how can we, work together to work with maybe philanthropic bodies and other private sector, private and public funding bodies to accelerate the funding and development of these systems. So that's another sector where we are currently working, and we would love to partner work with the patient support groups to kind of work together on this.
One last question I have is, as a genetic counselor, a lot of families would ask, is there a cure for the disease? Are there things that you can do? A lot of families are interested in registering for clinical trials and potential places where they can get access to research. So is there a forum or is there a place that we can, or like I know in the US there's like clinical.gov that actually helps you do this. So similar to that, are we creating any kind of forum or a website where we can at least start sending these people to register for that particular disease in the gene?
Right, right. Yeah, so right now in India, a lot of work in this space is still happening in academic labs. But then there are a few scenarios where they have now progressed to technology readiness level 7 or 8 or 9, maybe, where startups are beginning to develop some of these, model systems. But we are also building, we have a sister organization called Center for Predictive Human Model Systems, which is developing a database of all the labs and companies in India which are developing these systems. And hopefully, I mean, as the technology readiness level also increases, then they would have the opportunity to kind of be more participative in these systems. But then we do, I mean, right now that center does have a database of all the work that is happening, but we would also like to work with the government for them to have like a government database of all the work that is happening in this space. So that's also something that we're working towards, because like you said, they should have an opportunity, but right now we need more, a bit of more work to kind of get to that stage where we can use this data to, I mean, adopt for clinical trial stages. So
And the last question here is, how can we help the Center of Excellence to treat children who are dealing with rare disease?
sorry, can you just repeat that?
How can you help the Center of Excellence to treat children who are dealing with rare disease?
Right. So, I mean, another thing that we would ultimately like to visualize is that Center of Excellences also have dedicated funding for these model systems. Because right now, if there are centers of excellence have X percentage of funding, so out of that have at least certain percentage of funding allocated to develop these models. Because right now, most of the centers of excellence, they are not working on developing these human based model systems. So we need to have certain, again, a lot of it comes down to funding. Like they need to have dedicated funding to develop these systems, so in those centers of excellence, so that they can kind of help patients in a more direct way. So that's also something that we would probably have like a policy discussion with ICMR, and seeing how we can have like the Center of Excellence also having more dedicated research work in this space.
Thank you so much, Dr. Surat. This has been a very insightful conversation, and I think also quite motivating for all of us who work in this space to make sure that we all contribute in our own ways. Um, and we hope that there would be soon a lot more therapeutics that are available for children. Thank you so much for your time today. Thanks, audience, for your participation and questions. Have a good evening.
Thanks a lot for giving me this opportunity. Yeah, it's been a pleasure to speak here. Thanks.
Thank you, Dr. Surat. Thanks for this. Many people will not be able to attend, but they keep asking. It will help us to record this and send. In fact, now we are publishing last 1 year into one page link, so that it becomes a repository for every healthcare professionals and other things. Thank you so much.
Thank you.
Thanks.
Thank you, Dr. Prasanna.
Yeah. Bye.
Welcome.